Hello, everybody. Happy Friday afternoon. You guys are a very lively bunch. This is great. And there are very few people in the front row. It's very interesting, a little bit like church in that way. So, my name is Jonathan Lauer. You are more than welcome to call me Jonathan. I'm an assistant professor of physiology here at Marion. My primary teaching appointment is in the DO program. So I'll be seeing some of you in a couple of years, I'm very sure. These are members from my laboratory. I want them to introduce themselves. We're missing a couple of people, so if you can pretend to be somebody in addition to yourself, that'd be great. So your name and what you're doing while you're in my lab, maybe what kind of student you are, besides making bad life choices. Ending of my lab, no.
I'm Nick Weinstein. I already know a few of you from tutoring. Thank you, Lottie. I'm a research student in Dr. Lowry's lab, currently working on taste receptor signaling and bone remodeling.
I'm Krista. I also tutor, so shame on all of you that haven't come and met me. I'm a master's student in Lowry's lab, and I'm working on the BMPR2 pathway and identifying the gene signature which is as exciting as it sounds.
Hi, my name is Sylvia Hlebeck, and I am a second-year med student here. I joined Dr. Lowry's lab this summer to get involved with better understanding research, and was involved with targeting activin, which is a ligand, in terms of like bone absorption, and we're working on that still throughout this year. So yeah, looking forward to seeing where that goes.
Hey guys, my name is Tim. I'm a first-year student here at Marion. I also got into research with Dr. Lowry this summer, working on specifically the BMP and activin pathway. You guys will learn about that eventually. But that and skeletal muscle cells with respect to age and disease-related decline.
Hi, I'm Ruth Ann. I actually started working with Jonathan after my freshman year. I'm a junior now in undergrad, and I work with him looking at antibodies that can silence the ligand binding function of BMPR2, which is a receptor. So. Cool.
Okay, so we're missing Hindu, who I saw just a moment ago. Hindu is a second-year medical student. She's working on some biotechnology-related projects in my lab. So the reason one of these guys to come down front is, let me just tell you just very briefly, you guys, I promise you can go in just a moment. I am an educator scientist. My entire life is finding ways to connect students who are interested in research with research opportunities that can give them demonstrable outcomes so they can get where they want to go. So let me go through this list. Master's student expecting to do a PhD in the future. He needs publications. Krista Jackson, master's student moving to medical school, wants to do a competitive residency. She needs publications. Sylvia Hlebeck, second-year medical student, wants to do a good residency, needs publications. Tim Cho, first-year medical student, wants to do a great residency, needs publications. And this dear child here, we're not sure if she's going to medical school or graduate school, so I'm working my magic. I'm sending her good vibes on PhDs every single day. All these individuals are important to me because my life is helping them get to where they want to go. As it says here, I measure my success by their outcomes. This is my entire life, so I'm super excited to tell you about my lab today. I'm going to run through a couple things. You guys can please have a seat. Thank you for coming down front. My three goals, imagine this were a first date, and you and I had just met, we had just exchanged digits at some club. I'll tell you, this is how my wife and I met. We went on a date a few days later, and just straight out of the box, we're both in our mid-20s, and we were sort of done with the whole dating scene. This is well before Tinder, so it wasn't quite that bad yet. But on the very first date, we were like, look, you're not an axe murderer, and neither am I. Let's see how this goes on a second date, and let's lay down some ground rules with this. If you want to call me, call me. If you want to go on a date, say you want to go on a date. If you want to send me a text message, send me a text message. We were just so tired of all this dating game about, I'm interested, but I don't want her to know that I'm so interested, so she might run away too fast, these things. So my three goals for today, just being absolutely, completely clear with you guys here. I want to convince you that my lab is a fun place to train. I want all of you to leave here with that understanding today. I want to convince some of you to switch to the research track. My understanding is, at the moment in the room, there are next to none on the research track. And I hope to give you some little nuggets of understanding of why that might be a good decision. And those of you who do switch, I want to convince 1 to 2 of you to join my lab beginning in the spring semester next summer to continue the work that these guys are doing and do your thesis in my lab. Okay, now, this is my website. I'm going to point this out to you real fast. I'll go back to the last slide here. You're welcome to find more information about myself, my career training in the past, and what my lab does by going here, johnlowry.com. We're very active on Twitter in the lab, and you're welcome to call me on Skype. This goes to my phone. Some students do, so you won't be the first or only one who connects via that way. Okay, now, let me tell you a little bit about our training this summer. You can go on Twitter and see all these posts yourself. We are hashtag Lowry Lab, among other things like hashtag stuff Lowry says, which sometimes gets me in trouble. This was the lab at the last day of summer. We're missing Tim because Tim was on a cruise during this photo. We had Superlatives Day where we gave out awards based on characteristics of the people in the lab. So we get the lights down a little bit. I know I can't read these very well. Nick, here on the right, his says most likely to drive a minivan because he was dubbed the lab dad this summer. So this is because I took a couple of trips and he was always the one to call when the PCR machine needed to be sent back for servicing or the incubator started screaming in the middle of the night or something. Let's see, Krista is most likely to go cross-eyed because she stared at gene sets for 3 months straight, I think it was, Excel files. Still staring at that. This is a high school student named Logan, who's also not with us today. His is most likely to break out in song, if I remember correctly. He's very active in musical theater. Ruth Ann is most likely to brighten your morning. She always comes in with great questions, like she'll ask you these things even before you have your morning coffee. She'll say, so what are you excited about today? I'm like, the caffeine in this cup. And then, let's see, Hindu here, if I can remember this one. Yeah, I'll show you photos of this. This is most likely to break her nose in a yoga pose, and I'll show you why in just a second. She did not break her nose, I swear. Okay, and this is Sylvia, who is most likely to persevere because her project hit roadblock after roadblock after roadblock this summer. She kept coming back. So, we recently got back from a conference in Denver. This is a very famous statue right outside the Colorado Convention Center. This is Nick and Ruth Ann and Krista, whom you've already met. This is Laura Scherning, who's a third-year medical student who worked in my lab last summer. These guys all presented posters out at ASBMR in Denver. Got great feedback. I heard really great things. I will point out that there was one party night at this conference where you get 3,000 scientists and clinicians dancing. Someone in this photo led the conga line in front of all these professionals, and you can go on the hashtag Lowry Lab Twitter feed to find out who that was. Okay, so move forward. We implemented lab yoga this summer to keep us all fit because we are a musculoskeletal research lab. We know that exercise is important for your bones and your muscles, and so most of us are doing various forms of warrior pose, except Ruth Ann's doing something completely different in the background there. I think she's in wheel. And let's see, what else do we do? We got a lab mascot. This is the prairie dog, long story. We haven't cured osteoporosis yet, but we have a mascot now. And he has a little pirate flag with a skull and crossbones. So I think that makes us officially hardcore. Now, it's also important for us to celebrate success. I'm going to turn the speakers up just in case this plays as I hope it does. Let's see, this is not behaving. Move the prairie dog, he's in the way. Well, you'll just have to follow here. This might be line dancing in the hall after someone got a good Western blot result. Might be. Might be. Okay, so now we participate in a new venture here at Marion called the Bone and Mineral Research Group. Dr. Hum, your own Dr. Hum, and my lab get together for co-training opportunities. My students are her students are my students. We're all one big happy family. So we had a retreat together this summer. We set learning objectives for the group. You can follow our website here. It's tinyurl.com/bmresgroup. We're also active on Twitter. Krista Jackson manages that Twitter handle. She has a great sense of humor. So you'll find lots of fun things from this summer. This was the entire group at the beginning of the summer. It was big and a lot of fun. Okay, now, what do we actually do? This is the research landscape of my lab. We do lots of different things, but generally they fall under the level at biochemistry or cell biological up to physiology and usually trying to understand pathophysiology. We're always keeping our eyes open for a translational opportunity. Ideally, something bubbles up from the bottom, becomes something we can exploit as a possible translational approach. We're not going to be the ones to carry it to the finish line, but can we get something close enough that we can pass that off to a larger lab that has access to the right kinds of equipment, funding sources, those kinds of things is what we look for. We've also started doing a little bit of non-bench research. This is something that Dr. Sarah Zoll, along with Hindu and a second-year student named Connor Kirby, we're collaborating with 4 other colleges of osteopathic medicine to understand at the current moment, as we are facing a merger of AOA and ACGME residency programs. Those are typically DO residency programs and ND residency programs. You may not be aware those are merging. So there's a lot of concern at the moment about what is the level of research expectation of DO graduates. I see another lab member just walked in the back. So we are currently surveying at 5 different comms, the level of research experience of incoming first years, the level of research interest among first-year medical students, and then what might encourage those who are on the fence to do research, and what are some reasons why a person might not do research. And this is actually more difficult and more complicated than you might imagine. So we're pouring through those data, and we'll get them out very soon. Our comm is in great shape with regards to all those things. We're active in writing several review articles. This is a great way for students to get involved, where if there's a topic a student is interested in, we can most likely carve out a niche and find something interesting to write about and then publish that as a written project. We collaborate internally. This would be a collaboration with Dr. Michael Fontaine about eosinophilic esophagitis, which is an inflammatory-driven process. This is the project that Hindu was working on, where we're making biotechnology tools to evaluate cell signaling at the biochemical level. Nick mentioned he was working on nutrient-sensing GPCRs. Those are taste receptors. I'll talk about this project today. Mechanisms of skeletal muscle regeneration. This was Tim. He mentioned about sarcopenia and muscle regeneration. And then most of my work in the past has been on the BMP and active inner TGF-beta signaling pathways. We do this in the aging skeleton. We look at skeletal development, and we're also looking at translational approaches that will modulate this pathway. Okay, now, let me pause here and tell you, you can stop me at any time. I would rather not get through all my slides today, but we all end up at the same place together, then leave a few of you behind. Okay, so stop me whenever you have a question. I want to take a moment and do something that I was never exposed to in graduate school, which is someone just cut through all the niceties about how a research study began here because you had this incredibly important question, then you got a cell science or nature paper out of it. I was always craving someone to actually tell me the real story of how a project developed. Nobody ever talks about this. Nobody says, well, at this point, I didn't believe my data and almost threw the project in the trash can, but then 3 days later, something else happened. I want to tell you that story of our taste receptor project today. I want to be upfront with you about science development. My hope is that you have a better understanding of how students fit into this process, okay? Because it's not a one-person show. It's a collaborative process, and you'll see tons of collaboration today. My path to getting here at Marian began as an undergraduate at a research-limited university outside Jackson, Tennessee, in the middle of cow country. I entered as pre-med because I grew up in a small town in Alabama where if you were smart, you went to go be, if you were smart and good at math, you went to go be a physician because that's really the only thing you knew to become. Okay, so I went to college thinking, well, I'm good at science and math. I want to be a physician. That's my option. I decided to get a research internship at the Medical College of Georgia. This is in Augusta, Georgia. And to be absolutely honest with you, the reason I did it was to pad my resume for medical school. I had no research as an undergraduate. There just weren't opportunities. So I thought, I'm going to leave campus and go learn something new. That summer changed my life because I was suddenly exposed to research as a career choice. I'd never even thought of it before. I thought all PhDs were lab rats that never saw the light of day. And despite my pale skin color, I do get sunlight. This is actually a fun life. Okay, so I went back to college that fall, graduated just one semester early because of some CLEP credits and stuff, and had some time to figure out what was I going to do with my new bachelor's in biology. I pursued my med school applications simultaneously with my grad school applications. And then when grad school started looking very promising, I just never even finished my AMCAS applications at the time. So I ended up entering Vanderbilt University School of Medicine for a PhD in 2005. This is in Nashville. So most of my life at this point had been spent in the South. In 2010, I graduated with a degree in cell and developmental biology. During graduate school, I obtained a training grant, which was competitive at the time. It gave me funding. It also gave me a little bit of leverage to be able to say, well, I have this money, so I'm going to spend it this way. And that was liberating to be able to drive a project and do what I wanted to do. I also learned during that time in my life that I'm a teacher. And so I took 2 sabbaticals during graduate school to teach at the undergraduate level. I taught at Lipscomb University in Nashville, which is a private liberal arts university, and also taught at the community college level, which was an amazing experience. Because in that classroom setting, there were super high achievers who were trying to get ahead for the summer, and there were individuals who were going back for a second career choice. It was mostly nursing students in my class, and so I had those at the top that wanted more information than I could possibly give in a lecture, and I had those at the bottom who didn't quite understand what a nucleotide was. And so this is a great experience to learn how to teach to both ends of the spectrum. When I finished my PhD, I was offered a job in the Nashville area at Lipscomb, but decided to keep my doors open as much as possible. And so I took a job as a postdoc in developmental biology at Harvard. We moved from Nashville. I was already married at this point. My wife and I moved to Nashville with the dog in tow. It's a long drive from Nashville to Boston with the dog. We made it there, and I spent 4 years at Harvard. My postdoc advisor knew my love for teaching at that time. So about 3 weeks after arriving in her lab, I had an undergraduate from Harvard College assigned under me. I barely knew where the Western Blot room was, much less how to mentor this new student. So I was asking her a lot of questions. So to get to Fenway Park from here, where do you go? She's like, there, that way. So it was a great experience. We learned a lot from each other. Those experiences really taught me that I wanted to end up someplace like Marion. And so in 2014, I cast a very wide net for job opportunities. A couple of things worked out. I was able to join the Division of Biomedical Science here at Marion. And since that time, I've been working very hard to establish a research program that's founded on engaging students. Okay, so I've been doing a lot of career-branded thinking, and my tenure portfolio goes in about 11 months. So this term WIG here, W-I-G, that stands for Wildly Important Goal. You can barely be at Marion more than a couple of minutes and not hear President Elsner talk about the WIG. Our WIG at Marion is to have 2,500 graduates by 2025, I think is the WIG. If I mess that up, don't tell President Elsner. I should know this cold. My professional WIG is to exceed the minimum qualifications for promotion and tenure by next August. That is research, service, and teaching. So this is the right place. My career path is actually more of a choose your own adventure story. Some of you may remember the Goosebumps books from a long time ago. When you were in kindergarten, there were books called Goosebumps. Okay, so some of these would be you get to page 10, and you either go up the stairs or you go down the stairs. Down the stairs, there's an ogre, and up the stairs, there's a ghost. And the story just unfolds based on whatever your choice is on that page. So you may all be familiar with this kind of dendrogram where there are bifurcation points. You go left or you go right. And as you're sitting in this room, you are facing bifurcation points in your careers. Okay? Many of you want to go to medical school or some other kind of professional school. All of you are in the med school track at the moment, but there is a bifurcation point called the research track in front of you that you can go down the stairs to that ogre. Okay? My most recent bifurcation point was at MUCOM or somewhere else. I had a job offer at a school in Virginia. Looked great on paper. My wife and I sat down about it, looking at all the options. We decided that Indianapolis was the right place. Okay, so that was the most recent bifurcation point. Before then, it was choosing between a teaching-focused university or a research-focused university. This was a pretty easy one for me to decide. The lifestyle is completely different between the two, so I ended up in the teaching realm. Before then, it was a question of, well, is academia right? Or you can make a lot more money in industry, where you have a 9-to-5 position. If a project fails, you roll on to something else. Well, before then, I actually went to Vanderbilt to become a bacteriologist. I wanted to study Helicobacter pylori infections and their connection to gastric ulcers. When I got to Vanderbilt, the person I wanted to work with did not have funding for me, so I scrambled. I studied pulmonary vascular remodeling in pulmonary hypertension in graduate school, which is not a bacterial-mediated condition, okay? So that's a bifurcation point. I am now a cell biologist and developmental biologist instead of a bacteriologist. Before then, as I told you, at the undergrad level, it was physician. Before then, when I was a kid, it was an architect. I thought it would be great to be able to design big buildings that are beautiful things. And before then, I wanted to be a motorcycle gang leader. Okay, so this is getting well back in the past, at least a couple of years, right? The hours are terrible and you don't make a lot of money. Okay, so these bifurcation points, and you can probably think in your own life, you have these individuals that when you think about What led you to Marion? You can probably think of individuals who gave you advice along the way. These two individuals are my major mentors in my career. Mark DeCasticker is the gentleman here. He was my graduate school advisor at Vanderbilt, and this is Vicki Rosen, who was my postdoc advisor at Harvard. I still publish with both of these individuals. They are still very much part of my life. I wrote Mark a letter of recommendation about 2 weeks ago. He's going up for full professor at Vanderbilt, so we're still very much in contact. Vicki is on Krista's thesis committee. These are these interwoven, interconnected relationships that still exist. The other people that have greatly influenced my life are these students. I mentioned to you that my life is about student engagement. These are some of the students that, as I was making a decision to end up at the teaching university or the research university, I thought in my mind about these individuals. And going from the top down, this would be Eric Smith, who is a practicing dentist in the Hartford, Connecticut area now. He was in my lab, under my tutelage in Vicki's lab at Harvard. This is Annie Levine, who is now a medical student at Hopkins. She was an undergrad who was assigned to me before I knew really what was going on as a postdoc. This is Whitney Gao, who came under my tutelage at Harvard when she was a high school student from Arkansas. This is Jose Maria Amich Minero, who is now a junior at Harvard. He's from Barcelona, Spain. This is Alex Andonian, who is just finishing up this year at Bates College in Maine. These guys co-trained underneath me. They were a lot of fun to work together. Annie was under my tutelage for 3 years total. Eric was 1 summer. Whitney was 1 summer. And I still search these guys out to find out what they're doing. And I'm in email contact with them to find out how I can help. Okay, so these people were hugely important to me. You may be recognizing that there's sort of an up and down pattern to some of these kinds of decisions. I found in my own professional life that when things were going really well, I wanted to end up at a high-power R1 university where my life was driven by grants so that I could change the world. And then I would be reminded, sort of brought back the other way, what is the lifestyle I actually want? And you may be thinking about this yourself. What kind of medicine do you want to practice? Do you want the high-intensity 85-hour-a-week for the rest of your life? Or do you want to become a parent that spends time, maybe 3 days a week in the clinic, and you have the rest of the week off for your kids? Those kinds of things. These are the same kinds of up and down decisions that I've made in my career trajectory. It reminds me a little bit of what I've termed the research sine wave. Okay, so this applies to life choices. It applies to projects as well, and that's where we're going to go next. Projects, just like every decision, rise and fall. Great result, terrible result. Great result, terrible result. Over and over, you can just ask the lab. We had tons of those this summer. So for the rest of the talk today, I want to go through these things. There was an interesting observation. We decided what to do. We gathered and interpreted results. We had troubleshooting along the way, and we decided how and if to proceed, finally getting to the dissemination of results. Okay? So it began a little bit like this. In February of 2015, just after I'd arrived here that previous summer, there was a conference called Indiana Physiological Society that met here on this campus. Out in the atrium here at the Evans Center, I met this individual, whose name is Brian Dewar. He is now an associate professor at Taylor University in Upland. Is anybody from Taylor? Any Taylor graduates in the room? We occasionally get a few. So Brian is in biology there, and we had just met, and over lunch I said, hey, if there's anything I can do to help you out, we have some funding and some resources here that you may not have at your fingertips at Taylor. So a little while later, he said something along the lines of, there may be artistic license in this, but he said something like, hey, dude, I don't know well at all and haven't had the opportunity to decide if I do or do not trust. Do you want to collaborate? And I was like, yeah, sounds like a reasonable, well-constructed plan that can't go wrong. Let's do it. What will we work on? And he was like, I don't know. And I was like, I don't know either. And then a little while later, this happened in about June, after Mike Eaton, who's now a fourth-year medical student, was in the lab. He was there. He can attest to the accuracy of the statement. Brian said, hey, dude, I still don't know well. I found something interesting. And my response was something along the lines of woot woot or something like that. It was an acceptance of his offer, okay? So we then moved forward to evaluate this paper. This came out in PLOS One in 2013. Turns out I know one person on this paper. I didn't realize it until very recently. Erica Scheller, we interacted with last week or 2 weeks ago at ASP Mars. She's now at WashU in St. Louis. She's the one, Krista, who offered to analyze our bones at WashU. Okay, so this paper was sweet taste receptor deficient mice have decreased adiposity and increased bone mass. When Brian brought this paper to a lab meeting that day with his students and my students in tow, I thought taste receptors were only in your mouth. They're apparently not, okay? So what these authors found, and it's here in this blue box, is that T1R2 and T1R3 knockout mice have increased cortical bone mass and trabecular remodeling. Frankly, this was a little bit of a one-off observation in the study. They didn't provide any further information on why these animals have high bone mass. We were interested in it because there seemed to be some low-hanging fruit, a way for us to enter a new collaboration in a new field, new things we had never discovered before, and sort of make a foothold of being the taste receptor in bone lab. Okay, so we pursued that a little bit further. Here are the data. They showed that global deletion of T1R2 or T1R3, T1R2 knockout mice are on the left panel, and R3 are on the right, have a modest but significant increase in cortical thickness. This would be comparing the cross-sectional area of the tibia here of knockouts to wild types, or T1R3 knockouts to wild types. Now, I can see from your faces you're not impressed, and neither was I, because these are very small changes. It's on the order of 10 or 15% at best. But why is that important? Well, if any of you have a material science background, there's a phenomenon where the diameter of the rod greatly affects the bending strength of this rod. You can imagine bones in many ways like a material. Even a small increase in cross-sectional diameter will lead to an inordinate amount of bending strength. So for instance, if you double the diameter of a rod, you don't just double the strength, you increase the bending strength by 8-fold. So a 10% increase will be a potentially beneficial increase in strength of the bone. Okay, so we sat there at lab meeting that day, sort of scratching our head. Mike was there, had just walked in. We were like, okay, what the heck do we do with this paper? We had to make the decision, are these findings interesting enough for us to follow up and form the basis for future research? It was a decision of making a huge financial investment into this, a time investment into this, and students need papers. So putting a student on something that's going to fail or has a high likelihood to fail is often a risky proposition. So we had to say, well, if yes, then what will we do? Okay? So this is where we had to sort of evaluate, what do we know from the paper and what do we know from other sources? What do we know from Simon et al. And other sources? Well, we know that global loss of TAS1R3 or TAS1R2 results in modestly elevated cortical bone mass, and I will tell you about the biology of these receptors in just a moment. We know that TAS1R2 and TAS1R3 protein is quite widely expressed. It's not just in the taste buds. It's all throughout the alimentary canal, in the liver, in skeletal muscle. We now have demonstrated it's in bone. It's not just, taste receptors are not just involved in taste. Simon et al. Reported there's decreased adiposity in both of these mutant mouse lines. And from prior work, we knew that a mesenchymal stem cell's choice of becoming either a bone-forming osteoblast or a lipid-storing adipocyte are mutually exclusive decisions. So these authors in 2013 proposed the idea that since adiposity is decreasing these knockout mice lines, that maybe cells are being shifted to the osteoblast lineage that would form more bone instead. Okay? This is from a review article that I put out a couple of years ago showing that the taste receptors may be mediating this decision from a mesenchymal stem cell becoming an osteoblast or an adipocyte. Now, because of the technology that Simon et al. Had used, we already knew that knockout mice were available. You guys familiar with knockout mice technology? You delete a gene, sort of like if you want to know which part of your car steers the wheels, then you take off the steering wheel and then you can't steer any longer, then that's the implicated part. Okay, it's a knockout functional approach. Okay, now we were also able to infer that these heterozygous mice are fertile. That's important because if you're entering a new project, you have to think about, well, if I'm going to breed mice, how successful am I going to be in breeding these animals? It's not always possible that, it's not always the case that a heterozygous global knockout mouse is actually fertile. So getting homozygous knockouts can be a real challenge. Okay, and we also knew from other sources that the ligands are known for these taste receptors and that chemical inhibitors are available for this pathway. So I'm going to ask you guys then, what are G protein coupled receptors? These receptors, TAS1R2 and 3, are GPCRs. What is a GPCR? I'm going to draw this cell membrane here. How many passes does a GPCR have? People always know that. I love it. 2, 3, 4, 5, 6, 7. Okay. Let's call this T1R3. Okay, so what are the goodies on the inside, on the cytosolic side, that mediates G protein coupled receptors? G proteins, okay. Are they small G proteins? Are they heterotrimeric G proteins? Heterotrimeric, okay. And the 3 subunits are alpha. And I have no idea if I'm drawing this correctly, but let's put those there. Okay. Now, when ligand binds, these dissociate, right? Okay. Now, can you think of, with regards to alphas, there are a few classes of alphas. Can you think of what these are? G alpha Q. I hear a Q. Yeah, that's great. Does anyone know the downstream signaling events of a G alpha Q? Anybody think? Here's some mumbling. I'll help you out. It's typically calcium. And I think in terms of phospholipase C, there's several isoforms of that. Okay. What about some others? G alpha S. Okay, so that is what? Increases cyclic AMP, right? I think of PKA with that. G alpha, the other side, I. Okay. Okay. And then there's also the G alpha 11, I think is the other group. I tend to sort of forget about that one. Okay, now these G protein coupled receptors, the TAS1R3, belong to this group. Okay, so then if we put this here at the cell surface, 2, 3, 4, 1, 2, 3, 4, 5, 6, 7. Okay, these guys dimerized, 3, 4, 5, 6, 7. This is TAS1R2, TAS1R3. The ligand for this pathway are sweet molecules. Your favorite things to sweeten your coffee: aspartame, saccharin, sucralose. These hit the sweet taste receptor in your mouth. We presume then that the ligands are the same throughout the body. It is really hot down front. It's really hot over here. So, okay. All right, so downstream of this, I'm going to draw a squiggly line because there's a lot happening at the cell surface, but we know that there are at least a couple of downstream signaling events, like ERK1-2, and we know this activates the mTOR C1 signaling complex. Then you have all kinds of things downstream of this, like S6 kinase and S6, and then all kinds of squiggle stuff to eventually regulate protein synthesis. Okay, now, if these receptors are activated to induce protein synthesis or maintain protein synthesis, what do you think is the effect when the receptors are not engaged? Think for a moment. Why might these be present outside of the mouth where you don't actually need to taste sucralose or saccharin? What might they be doing? Any thoughts? Are you familiar with the process of autophagy? Have you covered this at any point? Okay, so can anybody explain autophagy? I see some nodding of heads. Anybody explain autophagy? Anybody? I could ask master student Nick Weinstein, who has his thesis proposal due on Monday, about autophagy. It's essentially a process where if the cell is in a nutrient-deprived environment, then it will start recycling its own components. It's deprivation. It's a deprivation signal. Okay, so the working model, and we haven't shown this yet in bone, but the working model in other systems is that these are surveillance mechanisms for surveilling nutrient load in the external environment. The major ligand is probably glucose for these endogenously. Okay. All right, so what might you do next? You don't have to answer out loud, but think through this for a moment. If you had this new paper, we had no reagents in our lab, no antibodies, no primers for PCR, not even knockout mice at our disposal. What might you do to enter this new field? What's the minimum number of experiments to decide if this is a good avenue or not? We ask questions like, well, what changes in cellular behavior would lead to high bone mass? Anybody with a physiology background, can you think through this? Okay, so what are the cells that make bone? Blast. What are the cells that resorb bone? Clasts. Okay, so which direction would this have to shift for you to get high bone mass? What would have more blast? How about like a seesaw mechanism when you were a kid on the playground? What if formation doesn't increase but resorption decreases? Okay, so it can be an accumulation defect. Okay, this is the same exact thing as osteoporosis. Just flip this in your mind the opposite way. Losing bone mass with aging is not because we necessarily form less bone, but it's because the seesaw is tipped the other way where resorption increases. Okay, so I tend to think about these things a lot like a seesaw. All right. Now, given the widespread expression pattern, how do we narrow down the mechanism? Okay, now think back to that automobile driving down the road. If someone were to say, I think that the tires are the ones that transfer the force from the axle to the ground, where might you expect to find the tires? Touching the ground, right? So you have to find them at the right place at the right time. So we thought about it in this terms. If we think that these are playing a role in bone homeostasis, we should find them in bone. And then we thought, what are the one or two critical experiments that if they fail, we cannot proceed? Can anybody think of one? Maybe there are probably several. Entering a new field, somebody else's results. What if we can't reproduce the project's dead in the water? Do you think? Nick, what do we have to reproduce? Right. We had to show the mice had high bone mass. Right. We would be chasing our tail on a dead-end project if we couldn't show the mice had high bone mass before we really got going. Okay? And then we thought, what will we need to build a publishable story, and really, how long will it take to get there? Okay, so then we came to the point where we had to think, how do we build a collaboration network that will help us get to this result? This is Brian up in Upland and myself here in Indy in our labs. We contacted one of Brian's friends over at Des Moines University named Eric Wasson. That's his picture on the left. Eric, lo and behold, happens to study taste receptors. He studies them in the heart. So he had the knockout mice. We were able to acquire serum from his mice. He shipped them from Des Moines to Indy. Brian's students came down to the lab one day. And these are the students that were involved: Mike Eaton, Jordan Newby, and John Arthur from my lab, and Maggie Plattis and Hannah Foster from Brian's lab. And we decided to jump in and start looking at what is the minimum thing we could do to decide this is a good idea or not. Well, so bone formation and bone resorption can be monitored via several biochemical markers. You can find these in urine, you can find these in blood. You can also find these in the tissue itself. We had serum, so we looked for a serum marker of bone formation rate, which is P1NP. This is the proform of collagen type 1. These levels tend to be clinically reliable for bone formation rate. And then we looked at the bone resorption side of things. Okay, so we got these data. You can look here. Where's the asterisk? On the right for the resorption side of things. So think back to this seesaw model where the mice have a modest increase in bone mass. We thought we were looking at a phenotype that wasn't due to an overt or exuberant bone formation, but a slow accumulation of bone mass because the osteoclasts weren't working as well in the knockout mice. Okay, so that was our interpretation. Bone resorption is reduced. We then started trying to think about where should we find these things? No one to this point had demonstrated that taste receptors were expressed in the bone microenvironment, so we took humeri from wild-type mice, fleshed out the marrow, ground up the bones, collected RNA, and did RT-PCR, which you're seeing here. We found that all three isoforms of the taste receptor type 1 family are expressed there. This is TAS1R3, 1, and 2, compared to a loading control. So we found that they are expressed in the bone microenvironment. We were able to look back, and in the supplement to a paper that Eric had put out in 2012, he had not actually demonstrated that they were present in the bone microenvironment, but that you could find transcripts in whole bone. It didn't rule out that they were present in blood that would have been found in the marrow. And I'll come back to the Kokobu et al. 2015 in just a moment. Okay, so now we began storyboarding. We had three pieces of information. We had the formation side of things, we had the resorption side of things, and we had the expression data. Three pieces of data does not necessarily a paper make, so we literally storyboarded. We do this in the lab all the time. And if you don't believe me, if you don't know what storyboarding is, it's where you take the data, you put it on little pieces of paper, and you tape it to things until it makes sense. This is exactly what you do when you write a graphic novel or anything else. We treat this like, if I was going to tell this story, how would it go? This is Nick and me sitting on the floor doing this on the door in the lab because Tim was doodling on the whiteboard that day and we couldn't, there was no room. So we were literally moving these things around in groups, trying to find the right story to tell. Okay, we do this all the time in lab. So what we decided was, well, we have these 3 pieces of data. It seems to make sense to begin with expression, then move to the formation markers, and then move to the resorption markers. But we had to find out what would link these things together. We needed something and something. Okay, so we had to decide what those somethings were. And it seemed to make sense that we would need to show that they were expressed in bone-forming cells and show they were expressed in resorbing cells. And if those things didn't work out, we could still make a story out of it. We just needed the data. Okay? So we decided to start branching things out a little bit. We had some cDNAs in the freezer and also acquired some primary bone marrow stromal cells from mice, and got those RNA. And we found that in the basal undifferentiated bone marrow stromal cells, these would be the progenitors for osteoblasts, we found T1R3 and R1. At that point, we thought, that makes sense. So these things would be dimerizing to not sense glucose, they're sensing amino acids. Okay, so that makes a lot of sense, which happens to be the umami taste, that MSG taste that you get with great Chinese food that's hitting that receptor. Okay, we did not find TAS1R2 there, but then we looked forward. You can imagine these are getting more differentiated the farther right you go. This would be an osteosarcoma cell line. But as you go right here, this would be a progenitor cell. This is an osteoblast-like cell, and this is an osteocyte-like cell. We found that T1R1 expression goes away and T1R2 expression comes on. So we had to switch our thinking just a bit. Maybe it's not sensing amino acids, maybe it's sensing glucose. Okay, so we had to interpret, though, that in light of our P1 and P data, even though taste receptors are expressed in this lineage, they're dispensable in that population. Okay, so we were forced with deciding, do we believe our data, both sets of data, are they interpretable, #interpretable data, and yes, we could make sense of those things. Okay, now, we didn't have osteoclast cDNA in the freezer, so we literally sent an email to Lillian Plotkin down the road at IU School of Medicine and said, Lillian, we need some osteoclast cDNA. Do you have any you can spare? She said, of course, come down and grab some out of the freezer, and we ran these, and lo and behold, T1R3 and T1R2 are expressed in differentiated bone resorbing cells in vitro, okay? We viewed this quite favorably. We thought, in light of our CTX data, that's the bone resorption data, maybe T1R3 is required in this population. We thought we had a good story. So we thought about then, well, fitting these things all together, is this a story that I would be proud telling and is going to be believable? If we begin the expression data and move all the way to the end, is this a coherent story? We felt like it probably was, but we had a few choices to make along the way. We could submit a paper straight away, and there's some pros of doing that as soon as possible. The students need publications. We need to move quickly. We might get accepted, or at least get comments that we could address and get in the revision pipeline. Or we might get rejected and end up waiting months to figure out what was going on. We could wait about the paper and continue building the story. That would mean that we're more likely to get published in a really good journal, but research in the research world, time is money. Moving fast is necessary sometimes, okay? We decided to go with option number 3, which is write the paper, but establish a track record by submitting an abstract to a national conference to sort of do both of these things, where we get the data out there and get comments. And so Mike Eaton, who is sitting at the back there, presented these. He wrote this abstract with me. We presented it along with the other authors at the American Society for Bone and Mineral Research meeting in 2015 in Seattle. We were the only people in this entire Congress studying taste receptors in bone. And so people would see the words nutrient sensor and come by and ask me about nutrition. And that's not this kind of project. It's a signaling project because there's this personification of taste receptors that they're sensing taste. But when you really think about it, they're a receptor with a ligand, and outside of the mouth, they're just a receptor with a ligand doing something else. Okay, so we had an uphill battle there. The data were not discarded, but we were the only poster about taste receptors. Okay, so we thought, well, we need to do something else. We need to bulk up the story a little bit, and we started scratching our head about the differentiation data here, where we had sort of a suspicion that the receptor expression shifted from R1 to R2 in osteoblast cells, and we wondered if the same kind of thing might happen in osteoclast. So we decided to firm up the story with differentiation assays. For this, we expanded our collaboration network. This guy is Aaron Brokey at the top there. At the time, he was at Carleton College in Northfield, Minnesota. I sent Aaron an email and said, Hi, Aaron. Haven't spoke to you in like 4 years. How are you? We want to collaborate, and we need somebody who has experience differentiating osteoclasts. Do you have any cDNA lying around, or could you do these experiments for us? He said, absolutely. Let me throw my student on here. And we're like, yeah, sure. Ryan's totally welcome. So Ryan and Aaron decided to start working on this. They acquired cDNAs from Aaron's postdoc lab, and we found that, lo and behold, TAS1R3 expression and TAS1R2 expression increase as cells become more osteoclast-like. So you take this precursor population, you differentiate them with the right goodies, they turn on TAS1R2 and 3. It was very supportive of our idea. The marker, the target on the right is cathepsin K. It's a marker of mature osteoclasts and is one of the enzymes that's used for resorbing bone. Okay, now, our working hypothesis then is that TAS1R3 is required for osteoclast differentiation and/or function. We felt like it was a pretty solid, if short, story. So we thought, well, now what do we do? Do we write a grant and submit a paper? Again, the same choices as before. Do we write a grant and continue building the story? We needed money at this point. And it turns out you write a grant, you submit a paper, and you continue building the story. So you do all these three things at once. And so this is what the lab set out to do. I decided to take a sidetrack with this, and I happened to have a Japanese colleague named Shuichiro Kokubu, the first author there. He and I did our postdocs together in Boston. Out of the wild blue, he happened to send me a paper one day about them studying taste receptors in muscle regeneration. And so we joined that. My name is the only Anglo name there on that author list and the second author there. And so we provided support for them to develop models for looking at taste receptor function downstream of muscle regulatory factors, which normally turn on things that are responsible for myogenic differentiation. This was important because I didn't want our paper to be the first one into this field for the lab. Coming in as someone who has a track record was important so that when we hit reviewers' comments, we already have a presence. Okay, so this was a very fortunate turn of events. And this got accepted very quickly. You can see we submitted, I believe it's cut off here. I think it was accepted within about 6 weeks' time of when it all started. So it was very fast for us. The students started writing the paper, and I sat down and thought, if I were a reviewer receiving this for the very first time, what problems would I have with the story? What might I ask for? Well, we mentioned independent confirmation of high bone mass, TAS1R family member expression in osteoblast differentiation assays, which we had not fully shown, we had sort of hinted at. Identifying the role of TAS1R3 in osteoclast differentiation and/or function, which we had sort of made marks toward but had not quite worked out yet. And then distinguishing, because these are global knockout mice, how do you distinguish a global phenotype that may be impacting resorption versus a resorption phenotype that's primarily due to osteoclast defects? Okay, this is the problem with global knockout technology. You have to narrow it down just a little bit more. I won't show you the data today, but moving forward with the lab, we did check this box off. We did confirm the mice have high bone mass. We have shown that that the expression does not change with osteoblast differentiation, like we originally thought. Nick's thesis project is performing this here, doing an in vitro cell-based model to work out the differences in differentiation versus function. And we're still thinking through the right way to ask the question of, is this an osteoclast-dependent defect? Is it cell-autonomous to the osteoclast population? Okay, so the very first time we submitted this paper, and I'm winding down here, we followed this tact, and this is normally how my lab does things. This is sort of when we storyboard, this is what we're going for. Build interest in the story by referencing the Simon paper and showing that these things are expressed in the right place at the right time. Rule out an obvious possibility by showing it's expressed in bone-forming cells and including the bone formation marker, which, if you find them here, you might think it's a bone formation defect. Support an alternative possibility here, and then leave something open for a sequel. So you sort of leave things at the end that you can build upon later. This reminds me a little bit of playing Texas Hold'em poker. You get your cards at the very beginning. You lay down the river, what's the beginning? You lay down 3, you see what's going on. You make your bet, you lay down 1 more, you evaluate again, you lay down 1 more, and you play your best of 5. We happen to think this was the best hand that we could possibly do as we put it into peer review. Okay? So we submitted June 30th of 2016. This is where we sent it to the journal. We submitted it to the Journal of Physiology and Biochemistry, which is an Elsevier title. Okay, and we continued building the story. Now, I want to show you here this sine wave. We've gone through several bifurcation points where at any moment in time we could have dropped the project. There was a lot invested, and it became increasingly more invested the longer we were in it. We have to get something out of it now. We've been working on this project for a couple of years. We have to get a paper out. Okay? So in February 2015, I met Brian for the very first time. In June 2015, we started working on this project, and we submitted the manuscript a year later. In the research world, that's pretty fast. There was a lot of up and down in between during that first year, but the ups and downs don't stop there. I've only shown you to this point. We submitted in June 2016, expecting to possibly get rejected, frankly. We didn't get rejected, but we were required to have a major revision, and this took about, 2 months. So time is money. We then resubmitted in January, thought we were going to get accepted. We made a taste receptor review article. I did this along with Sho Kokubu in Japan. Again, we talked about osteoporosis and sarcopenia, made a case for why taste receptors are important. And then we got reviewed again, and they wanted minor revisions. Okay, this is one of the most frustrating things that can possibly happen. You address the comments on the first round of review, and they give you more to satisfy. So I might have said some choice words. I might have, just don't ask my computer monitor what I said that day. Okay, so all summer long, we worked on revising this, and we resubmitted the manuscript. Just 2 weeks ago, Nick presented his preliminary data on the project at determining the difference in osteoclast differentiation versus function. The collaboration network has expanded again to be able to satisfy these reviewers' comments. Got good comments at the ASBMR meeting, and Nick is defending these data to his thesis committee on Monday. Okay, so this is where we are. What's coming next is we need to write a grant application in January to continue the project. Nick needs to defend his thesis in May, and then we need to submit a federal grant in June. Okay, so this is the rise and fall of the project. It was fast from the moment we got the collaboration going to when we thought we were going to make a story, but it has been a long and painful labor ever since June 2016. Okay, so we're now entering, what is this, 2 full years, 2+ years since we started this. This is what I wish someone had done for me in graduate school, where they told me about their coolest paper they'd ever done, and they told me all the points along the way where they cried themselves to sleep. Okay, these are the kinds of things in research where you have the bifurcation points of deciding, could we have stopped in August 2016 after we got the comments? Absolutely, we could have. But there were students who needed things, and we needed something out of the project. I want to quickly mention that there are a lot of people who put time and effort into this. The author list is pretty long on this project. Mike Eden, who's a 4th year, who is looking at doing a physical medicine and rehab residency next year, is the top author there. He's lead author on the paper. Jordan Newby was an undergraduate who came from my alma mater down in Tennessee to come to the lab for a couple of summers. Nick Weinstein, you met earlier today, who's continuing his master's project on this. Steve Shively is now a recent graduate who's an orthopedic surgery resident over in Ohio. Taylor Ward is currently a 3rd-year medical student. This is the rest of the lab who's been involved at some level in this project, although not necessarily directly in data generation. The collaboration was with Taylor, Brian Dewar, Des Moines, Eric Wasson, Aaron Brogy at Carleton, and Lillian Plotkin and her student Hannah at IU School of Medicine. We've been deeply helped by the Bone and Mineral Research Group with Julia and her trainees, and then this is the rest of the collaboration network that we're actively working with. My lab has been very generously funded with several faculty research development grants here at Marion, and also by the Indiana Academy of Sciences with a senior research grant to pursue Krista's master's thesis. Okay, now we have some time for questions. I want to stop here and answer any questions you may have. If you don't have any questions and you want to just go enjoy your weekend, that's awesome. Look us up on Twitter at my handle is at JW Lauer. You can do #LowryLab. You can go at BM Res Group and listen to Krista's musings. There's also a #BMRG. So, okay, what questions may I answer for you? Any questions? Yes. Could I ask you to use the microphone? Would you mind?
I like
Thank you.
Are there other taste receptors in the bone besides the family 1?
It's a great question. Do you have a background in taste receptors? It's a good question to think there might be other families. Okay, so you know the 5 tastes? Okay, so we've talked about umami and sweet. Bitter, sour, salty is the other ones, okay? Those are different families. That's the T1R2. I'm sorry, that's the T2R family. Okay, so we haven't looked at those exactly, and the reason is we haven't necessarily had a reason to go that direction. My suspicion is if they're in bone, they're doing the same type of nutrient surveilling, like environmental surveillance that the T1R family is doing. It's a good question to ask. The downstream signaling pathway is just a little bit different. So instead of activating mTOR, they go a different direction. We don't have the tools at the moment to do it directly, but we could get that way. Thank you for suggestion. Any other questions? Okay, well, my office is on the 3rd floor. I'm down the hall. Actually, if you're going to see Anea, stop one hallway short, turn right. I'm on your left. It's suite E, I think, is 317 is the suite. You're welcome to come by anytime. I keep office hours on Monday at lunch, and I keep Skype office hours on Thursday over the lunch hour as well. So you're welcome to hit me up either way. If I can do anything for you, please do let me know. All right, have a great weekend, guys.